Hi, my name is Francesca Ballesteri, and this is my exam mini video 5. A protein that is 4 amino acids long is formed from this DNA sequence in vivo. For this to happen, the DNA first needs to be transcribed into RNA. To do this, we need to determine which strand is the template strand and which is the coding strand. By flipping the bottom strand, we can see that both strands possess a start codon, ATG. The T becomes a U in the mRNA transcript. The coding strand will have a stop codon 4 codons down from the start codon. The UTG that the top strand codes for is not a stop codon, so it cannot be the coding strand. The blue bottom strand is the coding strand in this case because after switching the T to a U, we have UAA, which is a stop codon. Now that we have our coding strand, we can transcribe our mRNA sequence. To do this, we just have to change the T's to U's. Once we have our mRNA sequence, we can translate it to get our amino acid sequence. Our reading frame starts with AUG, which codes for methionine. We then have GUC, which codes for valine, AGC, which codes for serine, AAG, which codes for lysine, and UAA, which is the stop codon and does not code for an amino acid. If a mutation were to occur that changed a guanine to an adenine in the DNA coding strand, this same change would occur in the mRNA sequence. This would not result in a change in our amino acid sequence, however, because AAA and AAG both code for lysine. This is an example of a silent mutation because it does not change the amino acid sequence. If a transversion were to occur instead and the same guanine were to become a cytosine, the amino acid sequence would change because AAC codes for asparagine, not lysine. This is an example of a missense mutation because changing one nucleotide results in a change in the amino acid sequence. This change may or may not affect the function of the protein, depending on whether or not the affected amino acid is vital to the protein's function. Sticking with the same codon, if a transversion were to occur in the first nucleotide, changing an adenine to a thymine, which would then become a uracil in the mRNA after transcription, our codon that used to code for lysine now codes for a stop codon. We call this kind of mutation a nonsense mutation, and it would result in a shorter and likely non-functional protein. Here we have a transversion in the stop codon. An adenine has been changed to a thymine in the coding strand, which becomes uracil in the mRNA transcript. This would result in what used to be the stop codon coding for the amino acid leucine. Since the stop codon no longer performs its function, the mRNA will keep being translated until there is another stop codon in the reading frame. This is an example of a read-through mutation and would result in a long, non-functional protein.